Hi, I'm Allison Campbell. I'm the Associate Laboratory Director for the Earth and Biological Sciences Directorate. I'm here with James Stegen, who's a research scientist in the microbiology group. So James, tell me a little bit about yourself, where you come from and what you do.
Well, I actually grew up here and did my undergraduate and master's level work in Washington State, and then went to Arizona to do a PhD in actually large-scale ecology, theoretical ecology, studying patterns of species richness and linking that to the metabolism of organisms. And then went to University of North Carolina on an NSF postdoctoral fellowship, where I was linking large-scale ecological data sets with ecological and evolutionary theory. And then came here and switched from thinking very large scale to thinking very small scale, thinking about microorganisms and their impacts on biogeochemistry.
Cool. So we're standing out here on a beautiful day right next to the Columbia River, and it's not just happenstance. You do a lot of research involving river and the microbiomes and things like that. So can you tell me a little bit about that?
Sure. Yeah, we study the environment right along here, along the shoreline. And the reason is that there's a lot of material that is processed in those sediments that are just below the riverbed. Actually, about 90% of the metabolic activity in a riverine ecosystem like this is taking place right in those sediments. And so we're funded by the Department of Energy to study the fundamental biogeochemistry in those systems. A lot of implications for local scales as well as large-scale carbon and nitrogen cycling and contaminant fate and transport.
So you just published a paper recently, and you talked about the river's liver.
Mm-hmm.
And so the liver, as I understand it, is a big filter, right? Filters
Yeah.
our bloodstream of toxins and nutrients and all those sorts of things. So that's exactly what's going on here?
That's right, yeah. As the water moves into those sediments, they can clean a lot of the material, potentially contaminants or there's material that's in the river water. As it moves into those sediments, there's a great deal of microbial biomass on those sediments, and those microbes can consume and convert those contaminants or carbon and nitrogen pools and to cycle those or even retain or retard contaminant movement in those sediments. So it's been coined the river's liver because it's so active and it's so good at sort of cleaning and providing that ecosystem service.
How about if I ask what's the biggest difference from going from big to small? Are there differences, or challenges, or?
The biggest difference for me, so being a background in ecology, I actually think about the systems very similarly in terms of concepts, which is nice and I think powerful. But the biggest difference is really the scale of observation.
Mm-hmm.
So microorganisms, of course, are very small and we can't see them. And when we're thinking about like a tree community, we can go out there and we can directly measure a feature of a plant. We can measure its photosynthetic rate right there. And we can measure it, we can characterize its form and its function. But you're talking about individual microorganisms, you can't really do that, at least not at this stage. It's very challenging to get down to the scale of a microorganism and the scale at which important reactions are occurring. And that's sort of a challenge for the field in general, which is also exciting to sort of think about how to get down to that level and actually understand and study these fundamental interactions.
So you made a transition. You came here as a Pauling, a postdoctoral fellow, and then got hired on as a staff member. Any advice that you have for postdocs who want to transition into a staff member, whether it's here or wherever they go, on how to make that transition from a postdoc to a full-time employee?
Well, here I found it really important to not only just be open and very collaborative, but be willing to sort of put your ideas out on the table. And which can be sort of frightening to sort of put yourself out there and be willing to sort of be critiqued. But I found that to be a really important component of transitioning into a staff scientist and really a principal investigator.
Okay, well, thanks very much. I think it was awesome.
Yeah, thank you.
You bet.